hold of me on Twitter at onkarabetse_s. We left off yesterday. Yesterday, we're dealing with anaerobic respiration, and we also looked at anaerobic respiration in microorganisms and how it is used in industry. Okay, so just to recap what we dealt with yesterday, I'd like us to do a few questions. And guys, please use the group chat to send through your answers. And also, if you have any questions with regards to what we dealt with yesterday, just also send through your answers in the group chat, or alternatively, you can raise your hand. And also, please use the icons by participants just to give me an indication of whether we understand, whether we're together, or whether you are lost, and so on and so forth. Right. So let us start with the first question. Okay. It reads, is an adaptation that increases the surface area for respiration. A, smooth outer membrane. B, permeable outer. D, presence of DNA. So now this question is based on the mitochondria, if you are perhaps lost. And it's asking about the adaptation of the mitochondria that increases surface area for respiration. Is it the smooth outer membrane? Is it the permeable outer membrane? Is it the presence of cristae, or is it the presence of DNA and the ribosome? So we dealt with this, I think it was on Monday. Yes, we dealt with this on Monday when we were looking at aerobic respiration. Please send through your answers, guys. And if maybe you did not attend grade 10 life sciences, so I think you should be well versed in this topic. Come on, guys, please send through your answers. Is an adaptation that increases the surface area for respiration. A, is it the smooth outer membrane? Is it B, the permeable outer membrane? C, the presence of cristae? Or D, the presence of DNA and ribosomes? Okay, so guys, only one person has answered. Please send through your answers, even if you're guessing. Even if you're giving an educated guess, just send through your educated guess answer. Right. Okay, so I got an answer which says it's D. Okay, let us see what's the answer to question 1. The answer is C, the presence of cristae. Okay. So remember, look at the question. It says the adaptation that increases surface area for respiration. So now we're looking at surface area, right? And we know that DNA and a ribosome, they don't play a role in surface area. DNA is dealing with the genetics of the cell, right? So remember, DNA does not play a role in surface area. Okay. If we can just quickly revise this. Okay, so this is the slide that we looked at on Monday. It says the structure of the mitochondrion. This is a cigar-shaped organelle found in both plant and animal cells. It is surrounded by a double membrane. The outer membrane is permeable to allow oxygen and pyruvic acid to enter. The inner membrane is folded to form finger-like projections, and these projections are called cristae. These cristae increase the surface area for the process of cellular respiration. Okay, so I hope everyone understands this. And if you don't understand and you need me to go in greater detail, I'd like you to please indicate. Okay, now let us move on to the next question. Question 2. Is an adaptation that allows oxygen and pyruvic acid to enter the mitochondrion for respiration. Is it A, the smooth outer membrane, B, the permeable outer membrane, C, the presence of cristae, or D, the presence of DNA and ribosome? So guys, I've just touched on this on the previous slide. Please send through your answers on the group chat. Is an adaptation into the mitochondrion for respiration. A, smooth outer membrane. B, the permeable outer membrane. C, presence of cristae. Or D, the presence of DNA and ribosomes. Just gave you guys the answer and I went back to the previous slide. But come on, guys, just send through your answers. And if I am going too fast, please just indicate for me to go slower by the icons on the participants icon there. Right, awesome. So I got an answer, and yes, it is indeed B. So the permeable outer membrane is an adaptation of the mitochondrion, right, that allows oxygen and pyruvic acid to enter for respiration to take place. And the type of respiration that takes place in the mitochondrion is aerobic respiration, right? Are we together? Are we following? Yes, no, maybe, go slower, go faster. Okay, let us look at question 3. So during anaerobic respiration in plants, the products are A, ATP, carbon dioxide, and water. B, ATP, alcohol, and carbon dioxide. Or C, ATP, lactic acid, and carbon dioxide. Or is it B, ATP, alcohol, and water? During anaerobic respiration in sorry about that, guys. During anaerobic respiration in plants, the products are A, ATP, carbon dioxide, and water. B, ATP, alcohol, and carbon dioxide. C, ATP, lactic acid, and carbon dioxide. Or is it D, ATP, alcohol, and water? Please send through your answers, guys. All right, so the correct answer is B, ATP, alcohol, and carbon dioxide. Okay, so remember that the products of ATP, carbon dioxide, and water, we get that in aerobic respiration, right? And option C, which is ATP, lactic acid, and carbon dioxide, we get that in animal cells, right? And then the ATP, alcohol, and water, that is usually in yeast. Okay. Now let us look at the last question before we look into today's work. Bread is able to rise when baked because of A, anaerobic respiration, B, aerobic respiration, C, the presence of alcohol, or D, both A and C. Please send through your answers to the group chat. So guys, remember, these are the type of questions that you might come across during tests and exams, especially if you're writing common papers, right? Remember that multiple choice questions are usually 2 marks. Okay, so I'm just going through this with you guys in order to show you the different ways in which you could be asked the content of the work. Okay. So I will need everyone's participation just to see whether we all understand or whether I need to explain certain concepts again. Right, I've got an answer. So one said it's C and the other said it's A. All right, now let us see which one is the correct answer. Okay, so the correct answer is A. Okay, it's anaerobic respiration. Remember, bread, it uses yeast. Okay, so if alcohol is formed, that would be a byproduct. Okay, so it is not, the bread does not rise because of the alcohol. Okay, it rises because anaerobic respiration takes place. Okay, so I hope we all understand this concept of why bread rises when it is baked. Okay, remember we also said that while the bread is baking, the heat, it destroys the alcohol. Okay, if you guys can just also just quickly refer to yesterday's slides or yesterday's lesson. Okay, now let us move on. Okay, so yesterday we left off when we were talking about bread making. So today we're just going to continue with bacteria, right? Anaerobic respiration in microorganisms, and now we're looking at bacteria. So bacteria is used in the making of cheese and yogurt. The type of bacteria that is used in making these dairy products is called Streptococcus thermophilus and Lactobacillus bulgaricus. Okay, so you guys, you don't need to know these fancy names. Just know that the bacteria that is used in making dairy products is a lactic acid producing bacteria. Okay, just like the Streptococcus thermophilus and the Lactobacillus bulgaricus. Okay, so those are the lactic acid producing bacteria, right? So during anaerobic respiration, this lactic acid and this lowers then the acidic pH prevents the other types of bacteria from growing. So then we have an acidic medium that forms, right? And other bacteria, they cannot grow, right, within an acidic medium, okay? All right, so now let us look at how cheese is made. So the lactic acid producing bacteria is added to milk. This bacteria undergoes anaerobic respiration to release lactic acid. Then a protease enzyme, very important to note that we're using a protease enzyme called rennet, is added to the milk. Rennet causes the milk protein to coagulate. So this just means that it thickens the milk, right? Then the milk curdles into a solid curd. The curd is cut into slabs and a liquid is drained away from the curdling process, and this liquid is called whey. Then the curd is compressed into blocks. They are then placed on shelves to ripen, and the ripening process organisms. Okay, and then the longer the cheese is left to ripen, the sharper its taste. Then the flavor or texture of the cheese is dependent on the strains of bacteria used in their production. Right, so that is just how cheese is made. So what's important to note here in the making of cheese is the enzyme that is being used, which is rennet. Okay, and also the type of bacteria that is used. We are using a lactic acid producing bacteria. Okay, now let us look at the similarities between anaerobic and aerobic respiration, because Monday we looked at aerobic respiration. Yesterday we looked at anaerobic respiration. So what is similar between the two types of respiration? Okay, so the first thing to note is that both processes, glucose is a requirement. So glucose is a reactant in both processes. All right. The second thing to note is that in plants, both aerobic and anaerobic respiration release carbon dioxide, alcohol as byproducts. Okay. And the third thing is the main product of both types of respiration is energy in the form of ATP. So these 3 points, guys, they're very much important to note. Okay. Now let us look at the differences between aerobic and anaerobic respiration. So the first one, we have the aerobic respiration here on our left-hand side. First thing is that aerobic respiration requires oxygen, and anaerobic respiration occurs in the absence of oxygen. Okay. The second thing is that aerobic respiration, it releases more energy. We have a net gain of 36 ATP. Okay, and then in anaerobic respiration, it releases less energy, and we have a net gain of 2 ATP. Is that byproducts are the byproducts released in aerobic respiration is carbon dioxide and water. And then in anaerobic respiration, the byproducts are carbon dioxide and alcohol, right? So you can also add lactic acid in animal cells, right? Are we all following? Right, so now let us look at the comparison of photosynthesis and respiration. So I got this table here from the Via Africa Teacher's Guide on Snaplify. And I thought that it would be important for you guys to note it, because when we were dealing with photosynthesis, we also touched on cellular respiration and how cellular respiration might be similar to photosynthesis. Okay, because if we remember correctly, that photosynthesis produces glucose, right? And those tend to be the products, right? So now let us look at the comparison between photosynthesis and respiration. So photosynthesis, the first thing is that it is a process of anabolism. And remember, we said that anabolism is the build up. Okay, so it is the synthesis of organic materials. The second thing is that energy is obtained from sunlight. The third thing, it occurs in cells containing chloroplasts. The fourth thing is that light is essential So in order for photosynthesis to take place, we need light energy, okay, or radiant energy. The fifth thing is that oxygen is produced. Okay, so remember that oxygen is a product of photosynthesis and carbon dioxide is used. So carbon dioxide is a reactant in the process of photosynthesis. Remember that the plant gets its carbon dioxide from the atmosphere. It uses the light energy or the radiant energy from the sun, and as well as it gets its water from the soil. Okay, and then from there, photosynthesis takes place. Okay, and the seventh thing is that it is typical of all green process of photosynthesis. Okay, and then now let us look at cellular respiration in comparison to photosynthesis. So the first thing is cellular respiration is a process of catabolism. This is breaking down. Remember, cellular respiration, we use glucose and we break it down. Okay, if we just go back to glycolysis, the 1,6 carbon molecule of glucose, it is broken down into pyruvic acid or pyruvate, and we get energy from that process of breaking down glucose into pyruvate. Okay. The second thing is that energy released is energy is released and it is stored as ATP. Third point, it occurs in all cells containing mitochondria. The fourth point is that it occurs at all times in light or dark, right? The fifth thing is that oxygen is needed for aerobic respiration. The sixth comparison is that carbon dioxide produced by plants and animals, which this is where aerobic respiration occurs, and plants, it is anaerobic respiration. Okay, the seventh thing is that it is typical of all living organisms, aerobes and anaerobes. So all living organisms, cellular respiration takes place. Okay, so in all living organisms, cellular respiration will take place, whether it's aerobic or anaerobic, but it will take place. Right. Okay, so now let us move on to the evidence of cellular respiration. So basically, this is experiments. How can we prove that a certain organism underwent cellular respiration? Or how can we prove that carbon dioxide is indeed released during cellular respiration? So guys, at this point now, we are going to look at certain experiments that in tests and exams about investigative questions and experiments, right? So it is quite unfortunate that we can't really do them with you guys, but I'll just try to explain them. Okay, so in order to make this process of explaining the experiments and investigation easier, I used this Zoom in Life Science Topic 5 Respiration 11, right? And I got it from Snaplify. So you guys can also just get hold of this ebook and just go through it after the lesson. Okay. So all you need to do is just download Snaplify on your mobiles, and you can just Google this. Not Google, sorry, just search for this Zoom in Life Science grade 11 and go through the topic of respiration. Right. Okay, so the first test that we have is a test for carbon dioxide. Is carbon dioxide produced during cellular respiration? So remember, that would be our investigative question or the aim of the experiment. Okay, so remember, during experiments, we have our aim, we have our hypothesis, independent variables, dependent variables, and so on and so forth. The lesson of photosynthesis, and this is where I went into great detail with regards to answering experiment questions or investigative questions. Okay, so now let us look at the test for carbon dioxide, and we'll just start with reading this. Let me just get the cursor. We'll just start reading with this "Did you know" section here. It reads as follows. Soda lime or sodium hydroxide absorbs carbon dioxide. So in the air, so that the air contains no carbon dioxide. And then we have another bullet there which says lime water or calcium hydroxide tests for the presence of carbon dioxide. So that is very much important to note. If you guys also remember that we also used lime water or calcium hydroxide when we were testing for carbon dioxide during photosynthesis. Okay, so now this is how the setup of our experiment would look like if we were to test for carbon dioxide. So we'd have two test tubes, okay? So one test tube would be our experiment test tube, and it is the test tube on the left. And the other test tube would be our control test tube. So what do we have in the experiment test tube is we have the stopper, okay? We have the muslin bag, which is suspended by a thread. So that thread is basically so that the seeds, the germinating seeds which are in the muslin bag do not touch the lime water, which is poured in the test tube. As you guys can just see here at the bottom, we have lime water in there. We have the germinating seeds, which are inside the muslin bag. All right. And all of these materials, they have a reason as to why we're using them. And I'll just go through it with you guys now. And then in the control test tube, we basically have our stopper, the same one that we have in the experiment. We have the same amounts of lime water, which is in the experiment test tube, but the only difference is that we don't have what? We don't have germinating seeds in the muslin bag. Okay, so I hope you guys can see that. So the use of having the stopper is that it prevents the carbon dioxide from entering the test tube. So that would prevent any atmospheric carbon dioxide from entering the test tube. Okay, and then we have the germinating seeds in the muslin bag. So guys, first thing to note is that the muslin bag has holes in it, all right? And those holes are so that the carbon dioxide that is produced by the germinating seeds can be released into the space of the test tube, all right? So now what should happen in this experiment is that the germinating seeds should undergo cellular respiration. Okay, and therefore, if the germinating seeds undergo cellular respiration, that means carbon dioxide will be released. Okay, and if carbon dioxide is indeed released, then the lime water will turn milky. So we would leave this experiment for a couple of days, 2 to 3 days, and then would come and check the color of the lime water. So what we expect to see is that in the experiment test tube, the lime water should turn milky. And then in the control test tube, this test tube here, the lime, then we'd know that carbon dioxide was present. Okay, so lime water changes color in the presence of carbon dioxide. It turns milky. So that is how we would prove that indeed cellular respiration releases carbon dioxide, because our lime water would have turned milky. All right. So just a quick question, guys. What would be our independent variable in this case, in this type of experiment? What would be our independent variable? What would be our independent variable if we're testing for carbon dioxide? Just send through your answers to the group chat. What would be our independent variable? Guys, if you do have any questions with regards to the test of carbon dioxide, just also utilize the group chat to write down any questions, or you can alternatively raise your hand. Okay, so I've got an answer, and I would just like to make a follow-up question, all right? So independent variable, remember we said that this is the variable that the scientist or the learner has control over. Okay, so this is the one that you, as the person conducting the experiment, you are changing, all right? So you have control over it. Okay, so the answer that I got was, is it lime water? Remember we said that we're going to leave this experiment for a few days. Okay, so time in this case would be the independent variable, because the lime water will change to be milky, right? If time is extended. For example, if we leave it for 2 days, it will be more milky. All right, I hope I'm making sense. So the intensity of the color change is dependent on the time. Okay, so therefore, time is the independent variable, right? So it was not far off. Your answer was not far off, but it is time. So time in this case is the independent variable, right? Now, let us move on to the next experiment. Testing for heat. Okay, and the reason I wrote energy in brackets is because, guys, remember, energy comes in different forms, right? And even when we are forming ATP, we also have heat, which is formed, which is released. And remember that heat is also a type or a form of energy, thermal heat, right? Or thermal energy, rather, sorry. So let us start by reading this did you know box here. It says the control will have one factor missing that is present in the experiment. The control is present so that you can compare the experiment measurements to the control measurements to ensure reliability, right? So here we have inverted vacuum flasks. So on the left-hand side, we have our control flask. Okay, right here we have our control flask, and here we have our experiment. So we'll be comparing our experiment to the control. Okay. And what we have here is a vacuum flask. We have the narrow range thermometer. In the experiment flask, we have germinating seeds, but in the control flask, we have boiled sterile seeds. In both the flasks, we have rubber or cotton wool to close the flask at the bottom in this manner. All right, so firstly, the flasks are gaping, okay? Because remember, guys, hot air rises, right? So if the flasks are inverted, the hot air will go to the top of the flask, right? And also, at the bottom here, it will allow the carbon dioxide, which is formed, to exit the flask, all right? And then in the experiment flask, we have germinating seeds. So we need germinating seeds in order to test for cellular respiration. All right, so cellular respiration needs to take place, and that is why we have germinating seeds in the experiment flask. And in the control flask, we have boiled sterile seeds. So we have killed the seeds so that cellular respiration doesn't take place. Okay, and then we have the rubber or the cotton wool at the bottom of the flask. As I said, this allows for of oxygen. Okay, so what we will see from this experiment is that the thermometer in the experiment vacuum flask will show an increase in temperature over a few days because heat will be produced by the respiring seeds. Okay, remember that respiration, when it occurs in seeds, we have the products that form are ATP, or heat or energy. And in this case, it will be energy in the form of heat. Okay. And we also have carbon dioxide, which forms. All right. So I hope everyone understands the test for heat or energy. So I'd just like to ask a quick question. Just send through your answer in the group chat. If I would ask you to write down a hypothesis for this type of test, how would you put it? Write down a hypothesis for this type of experiment. A hypothesis for this type of experiment. Write down a hypothesis for this type of experiment. It need not be long, guys. Just a brief statement. A brief statement. Remember, that is what our hypothesis is, right? A brief statement, or which is an assumption of what would take place in the experiment. So if you were to come across a question which says, write the hypothesis for this experiment, what would you say? Remember, our aim is to test for heat, okay? Our aim is to test for heat or energy. So what would your hypothesis be for this type of experiment? Okay, so as I'm waiting for answers, I'll just move on to the next test that we will do. And this is a test with yeast. Right. So remember we said that yeast undergoes aerobic respiration, right? So yeast undergoes anaerobic respiration. It's a type of setup that would have if we are testing whether anaerobic respiration really takes place in yeast. It says, for the control, set up exactly the same apparatus, but remove the yeast cells from the sugar solution. This will prove that presence of the yeast cells. Okay, so in this, but we don't have the apparatus for the control, but sorry guys about that, it was just a network error. As I was saying, that if you were to set up a control, right, apparatus for this particular experiment, you would not add yeast. So, okay, what do we have in this particular apparatus is that we have a thermometer, okay, and that is to measure the temperature. We also have the water bath, all right? And we're basically measuring the temperature of the water bath, okay? And we have liquid paraffin, this particular test tube here, we have liquid paraffin, and at the bottom here of in this first solution, to a glucose solution, which has yeast in it and water. Okay, and it also has a stopper, right? First of the liquid paraffin. So this oil network is not playing with me today. Sorry about that. Sorry. As I was saying, guys, that the oil layer or this liquid paraffin, it forms an oil layer which prevents oxygen from entering the glucose and yeast solution. All right, because remember, we're testing whether anaerobic respiration takes place. Okay, and then in this glucose yeast solution, we know that we put them together so that we test whether anaerobic respiration takes place. And we have a water bath there so that our apparatus is kept at a constant temperature, and we can measure that using our thermometer. And then these rubber stoppers here on these various test tubes are there in order to prevent any atmospheric carbon dioxide from entering the various test tubes, okay? Because that will in turn invalidate that we are supposed to say should be produced by the yeast cells. So carbon dioxide will be produced here, and the gas will in turn move in this particular tube into this test tube, which has lime water. And once the gas bubbles into the lime water will turn milky. And that will be an indication of indeed anaerobic respiration took place because carbon dioxide was produced. Okay. Right. So can move on. If you guys have any questions with regards to any of these experiments, just please write them down in the group chat. And if I can move on, just please indicate quickly for me using these participant icons. So do we all understand? You can just click yes or no. And if you need me to go through the dependent, independent controls, methods and materials, hypothesis, discussion, Shandis, just also please indicate for me. Okay, so we just have 1 minute. And I think we'll just go through the terminology tomorrow and all the questions tomorrow, because time is not on our side. But I would like you guys to just do this question here for homework, all right? So just please take a screenshot of this question and please do it this afternoon. And then in tomorrow's lesson, we're gonna go through terminology and the particular slide, okay? So just please take quick humans, study the diagram and answer the questions. So guys, I got this question from a post-comment paper, all right? It was a departmental paper, okay? So we have phase 1, phase 2, phase 3. Question 2.1.1, identify phases 1, 2, and 3. Question 2.1.2, name X and Y. Question 2.1.3, State the origin of X and Y. Question 2.1.4, identify products 1, 2, and 3. All right, so I hope everyone has taken a screenshot. And guys, just a quick reminder that today we have the Wednesday career advisory session. Okay, you guys can get the link to the meeting on the newsletter. All right, so the link to the meeting is attached to the newsletter at this end. All right, so just remember it starts at 3. It's from 3 to 4. Okay. Otherwise, thank you very much for attending. If you guys have any questions, please do not hesitate to send me an email. And also please follow Africa Teen Geeks on all the various social media platforms. Good day and goodbye. Enjoy your afternoon, guys. Right. Bacon, you have an answer?